Entonces, what are stem cells? It's a complicated issue. By definition, is he translating? No? Yes, he is translating. The definition of a stem cell is a cell that can self-renew and that can also give rise to cells of multiple lineages. So how do I explain this? Oh, sorry. Oh. F5 probably, right? No. Okay. How do I explain this? A stem cell is a cell that can renew itself. So if it's a hematopoietic stem cell, it can give rise to another hematopoietic stem cell. And do I have a pointer here? This is the pointer. So it starts as a stem cell, and it can turn into a second stem cell. That is self-renewal capacity. But a stem cell can also give rise to a cell that is more mature. A hematopoietic stem cell can give rise to a myeloid progenitor or a lymphoid progenitor, and then that progenitor itself can divide again into an erythroid, red blood cell progenitor, a white blood cell progenitor, or a megakaryocyte. So that is the definition of a stem cell. A cell that can give rise, sorry, I do something wrong here. A cell that can give rise to itself, it can self-renew, auto-renovation, but it can also divide in an asymmetric fashion. I apologize. In an asymmetric fashion, it can become different types of cells, and it can give rise to cells of multiple lineages. Now, a stem cell can be a very primordial stem cell, an embryonic stem cell, the cell that gives rise to all of us, but it can also be a cell that arises from a more differentiated tissue. There can be a liver stem cell, there can be a bone marrow stem cell that gives rise to hematopoietic cells, there can be a mesenchymal stem cell, and I will talk briefly about all of these cell types. Others will talk more about their applications and about what we can do therapeutically with them. I will give only very brief examples. I will try to introduce you to the topic here. So I divided the talk into 4 different categories. There are embryonic stem cells, iPS cells, which I don't know how to translate in Spanish, induced pluripotent stem cells. There are the adult stem cells, and perhaps a little bit different category are the cells that are derived from cord blood that we typically think of as hematopoietic progenitors, but that may be a little bit more complicated. So what are embryonic stem cells? Celulas madres embryonarias. They are derived from the inner cell mass of a blastocyst. So this is a 1-day or 2-day-old embryo, and one can pick out the cells from the inner cell mass that in nature will give rise to a human being or to a mouse or whatever origin it is derived from, and one can, for the last 20 years, put these cells in colonies and grow them in a dish and derive tissues of various organs or progenitors of various tissues from this. This is a very powerful discovery. Obviously, to me, almost as a doctor or as a biologist, it makes sense that this cell exists. We clearly all derive from an embryonic stem cell. But the fact that this cell can be cultured in a colony and then be manipulated to give rise to various tissues, that it can be genetically manipulated, gives both great excitement and opens all our eyes to great potential for regenerative medicine, because in principle one can grow anything from that. One could grow nervous tissue, one could grow muscle, one could grow blood. At least in principle, one could do this. One also is concerned about the ethical implications. What can one not do that could have unimaginable consequences? And this concept of embryonic stem cell colonies give rise to a lot of debate during the Bush presidency, as I recall, in the United States. I was already in the United States at that time. This is a very controversial cell, and it became, there are many ethical problems or questions, depending on what side of the debate you are on, and its use has been somewhat restricted, and even its research has been somewhat restricted. So how do we get around this? How can we create a cell that is similar to the embryonic stem cell, but that does not have the same ethical problems? There were a number of attempts, and you may remember Dolly the sheep. A lot of you will remember Dolly the sheep. And Dolly the sheep was derived from an egg cell, an oocyte, where the nucleus had been removed and another nucleus of another sheep implanted, and that gave rise, they took the nucleus from adult tissue of a sheep and they implanted it into an oocyte, an egg cell of which the nucleus had been removed. So they took the cell nucleus with the genetic information from one animal and implanted it into the environment of an egg cell that gave rise to the adult animal. And that worked, but never was much advanced. And about 10 years ago, so this, going back a little bit, this embryonic stem cell could give rise to pretty much every cell in the body. About 10 years ago, the concept, a group in Japan, Dr. Takahashi and Yamanaka, and Dr. Yamanaka subsequently received the Nobel Prize for his discovery, discovered that we could recreate an embryonic stem cell from an adult cell. One could take, I believe in their initial experiment, it was a fibroblast, therefore an adult cell, and by genetically manipulating this cell in a rather simple fashion, they knocked in 4 genes. The genes were called OCT4, SOX2, KLF4, and c-MYC. The most famous one in my mind is c-MYC of all these genes. All these genes are involved in early differentiation of embryonic cells. When they put these genes in an adult cell, then the adult cell acquired the properties of an embryonic cell. So they could de-differentiate an adult cell back into an embryonic cell, not a real embryonic cell that came from could come from a red blood cell progenitor, could come from a skin cell, could come from a fibroblast, and they could give it back these pluripotential properties. That was then called an induced pluripotent stem cell. And that really has revolutionized the field, because obviously there are no ethical problems with taking a cell from the skin or a cell from adipose tissue or a cell from any organ and reprogramming it and giving it back these primordial characteristics, these properties that allow it to de-differentiate into an undifferentiated cell and then re-differentiate under our command, let's say, into a cell that has specific properties. So imagine somebody who had a sarcoma and had an amputation of the bone, and it's a young child, and you have problems with bone growth for the rest of his life. In principle, at least, you could take cells from that person's skin, de-differentiate them into induced pluripotent stem cells, and grow bone. You could take somebody who had a car accident and had spinal cord injury, take cells from the red blood cells, de-differentiate them, and grow them back into neuronal stem cells and cause the healing of the nervous system. And I truly believe that's where we are going. But I think we are 10 years away from that, at least. And others will talk about that in much more detail. So, but that is where all the excitement comes from. So the principle is established. The practice is far from established. And I hope that others will talk about that much more. A lot of people are working on achieving these goals. And I will just give you one quick example of a recent publication where macular degeneration of the eye, a disease of the elderly, where cells of the retina degenerate, where they took cells from the fibroblasts of an elderly lady, they reprogrammed them with these 4 factors, induced them into induced pluripotent stem cells, re-implanted them, and this led to improvement in eye function. And I think this will be repeated again. This is the first, to my knowledge, practical example of clinical use of induced pluripotent stem cells. Now, that is fantastic. Keep in mind also the eye is an organ that is protected from the immune system. It's the easiest organ to do any type of genetic manipulation from and to change tissues in. And I believe they treated only 1 patient. And so therefore, this is very early days. All the other applications are years away, and this was done in a very careful fashion. But I think this is the future. Now, perhaps easier, let's take adult stem cells. What are adult stem cells? Adult stem cells are tissue restricted. They are multipotent, they are not pluripotent, meaning that they can give rise to their progeny. A hematopoietic stem cell can give rise to myeloid cells, megakaryocytes, red blood cells, but it cannot go back and cause brain tissue, or it cannot go back and create bone tissue, at least not in principle. The dogma is it cannot, unless we transfect it and make iPS cells, which is technically still very, very difficult. There are other stem cells. Every organ turns out to have stem cells. This concept really derives from hematopoietic stem cell transplant, where it has been known for many, many years, and we routinely transplant these cells for bone marrow transplant and for curing leukemia. But in the last 10 years, there's an explosion of knowledge on other types of stem cells. Gastrointestinal stem cells were found in the bottom of the crypts of the gastrointestinal system. Muscle stem cells exist. Pancreatic island, and perhaps Dr. Wickrama will talk a little bit about that. Mesenchymal stem cells. In principle, there are stem cells for every purpose. Other than using them for bone marrow transplant, hematopoietic stem cells, and a little bit for pancreatic islet transplant, I am not aware of that many applications, but others will talk about that. But there's a big question here. Is this dogma true that adult stem cells cannot differentiate in other tissues? And there's for a long time been a question, can stem cells, adult stem cells, convert into another type of stem cell? After all, in the plant world, this is not uncommon. One can put a leaf of a plant, some trees in the soil, and grow a totally new tree from it. So in a plant, a cell can differentiate and redifferentiate fairly easily. Does this happen with adult stem cells? And there have been long, intriguing observations. For example, in bone marrow transplant, occasionally we find like A year later, in a patient, we do a skin biopsy, and we see in the skin some cells that look surprisingly as if they were of donor origin. And the way we see that is by sex mismatching. We have transplanted a male with his sister, and all the hematopoietic cells are from the female. However, once in a while you come across reports where it is mentioned, listen, we found a cell in the skin and it looks like a true skin cell and it is of donor origin. So perhaps under the right circumstances, an adult stem cell can convert back into a cell of another tissue, be it to a limited degree or be it to an extensive degree. And that's, I think, where a lot of enthusiasm comes from, but also where a lot of the misuse comes from that was already mentioned. Because if that were the case, and we could do that reliably, then it would be very easy to grow new organs. It would be very easy to repair the spinal cord, because we would take bone marrow stem cells, very easy to obtain. We've done it for 30 years. We would inject them in the spinal cord and the spinal cord would heal. The problem is it's not so easy. And even though questions there continue to linger and a certain amount of conversion may be possible, it is probably extremely limited. And again, perhaps others will talk about that. But perhaps the most studies have been done in heart disease. And some of you may remember, these are already old studies I'm showing here. When preparing this talk, I thought back about the old days. And if you look back to the early 2000s, there were a lot of trials of injecting hematopoietic progenitors, what we call CD34 selected stem cells, either into the bloodstream or into the coronary arteries of people with a myocardial infarction, with the idea that we would hasten healing of the myocardium. And this is a paper in Nature, a very prestigious journal, I would say, of a mouse model where they injected GFP-modified hematopoietic stem cells in a mouse that had an artificially induced myocardial infarction. So they ligated the coronary artery. The mouse had a thinned myocardial wall, like it should happen if you have a myocardial infarction. The myocardial wall becomes fibrotic and becomes thin, and they injected hematopoietic stem cells into the bloodstream, and these hematopoietic stem cells were stained green. They were GFP, green fluorescent protein, and as you see, they show up in this infarcted area. So they are attracted to this infarcted area, and at least in this model, this is an animal model, mind you, this is not humans, but in this model, they hastened healing of the myocardial infarction. So there was a lot of enthusiasm about this. We are going to treat myocardial infarctions with hematopoietic stem cells. And there were at least 5 trials. And some of them were in the New England Journal of Medicine. And the results are inconclusive. So it's not totally nonsensical. It makes some sense. There may be something there. But if there's a capacity for reconversion and for healing other tissues, it is quite limited and may not be clinically applicable and may not have clinical use. And at least for myocardial infarction, I don't think we are doing any studies any longer. 10 years ago, I remember that I was working with Dr. Wickram. I remember that we talked to our cardiologist, should we initiate studies about this? But that field, although initially with a lot of promise, seems to have failed. Now, that has not stopped people from doing this. And there's a lot of studies, a lot of clinics around the world where people take cells from any tissue and tell their patients, we are going to inject it in another tissue, and your dementia will get better, your eyes will get better, your walking will get better. And that is the problem. This is published in the New England Journal of Medicine in 2017, I believe. And this was a clinic in Miami, Florida, where patients came with macular degeneration. And instead of this careful study that I showed you a couple minutes ago, where one patient was treated in a very complicated fashion, no, they took adipose tissue. They took adipose tissue from the patients, and they did something to it, whatever it was, and they injected it in their eye. And there were 3 patients who were treated in this private clinic, and they then came to the University of Miami when they went blind. And so this, the University of Miami published this as a warning, and only then did the FDA crack down on this clinic, and this clinic is closed, I believe. But so this type of approach is what is the big concern about regenerative medicine. And the last quick point that I want to make, if adult stem cells can convert, a lot of people are putting a lot of hope in using umbilical cord blood stem cells. So we collect routinely umbilical cord blood at birth in a number of centers, and these umbilical cords contain hematopoietic stem cells. Adults have the hematopoietic stem cells only in the bone marrow. Newborns have them in their blood. And it makes sense that these cells, they are just newborn. They are literally a day old, or they have just had intrauterine life. They have very different immunologic and hematopoietic properties. They have to last as a lifetime, not another 10 or 20 years. And they are, at least in hematopoietic transplant, they have fascinating characteristics. A lot of people believe that they may have better characteristics of conversion into other tissues than adult stem cells. And they have been used in studies of cerebral palsy, where kids are born with cerebral palsy and get an injection of umbilical cord blood stem cells, spine repair, diabetes mellitus. Just from a review in the Transplant Journal this month, the number of patients treated on such studies is up to 800. The number of indications, neurological, diabetes, cardiac, and vascular, a number of studies. But the conclusion, once again, is these are studies. There's nothing established. There's some hint that this may work in cerebral palsy and alleviate cerebral palsy, but it is very, very early days in that respect. So at the end, and I borrowed a lot of these slides from one of my colleagues at Cornell, a PhD researcher who is interested in stem cells, Dr. Todd Evans, who I should acknowledge. And this was the last slide he gave me. And these are his words. Very few stem cell treatments are proven safe and effective. And I think we'll end up talking a lot about this the next couple of days. Clinics peddling unproven stem cell treatments frequently overstate the benefits and use patient testimonials to support their claims. An experimental treatment you pay for is not the same as a clinical trial. Cells from our own body are not necessarily safe as a treatment. That's also the statement of some of these clinics. You no longer need donor cells. We take your own cells. But if they are manipulated or if they are injected into an organ where they don't belong, they're not necessarily safe. They are less supervised by the FDA. They are less regulated. That is for sure. Park Avenue stem cell at the front. This is from an ad. It doesn't come through very much, but it talks about Park Avenue stem cell is at the forefront of regenerative medicine. Really. So with that, I thank you for listening to me, and I will be happy to answer any questions.